Is on the way. I'm Veronica. I work for PhotosynQ. I live in Ithaca, New York, however, so I spend a lot of time working with Cornell. And when I first got there, we were talking about Northern Leaf Blight and really thinking about how to use PhotosynQ with Northern Leaf Blight. So I'm going to present on determining early detection of Northern Leaf Blight. So Northern Leaf Blight is a fungal pathogen, and it forms large photosynthetically inactive lesions in corn. So the damage comes through the fungi's infection, feeding, and toxic metabolites. This causes yield loss, and it happens early on. So before silking, before tasseling, basically the plant hasn't had enough time to get starch or sugars for grain filling, and then you see yield loss at the end. Another part of having these huge damaged lesioned areas is lodgings, really common to occur if the infection is severe. So one of the things with Northern Leaf Blight is you kind of know because it's fungi, it's going to happen in wetter areas, it's going to happen in low-lying areas. But when you suspect it's happened, there's not a good, 5 days later you should go out into the field. Basically, if you have an idea, you just have to scout from 1 to 2 weeks for these lesions. If you can see it, that's a pre-lesion before it actually goes into a full-blown one. And then by there, you can say, okay, you need to apply fungicide. But they don't have good guidelines on fungicide application, when to actually make the decision. So it is pretty variable and on a case-by-case basis. For that reason, we kind of got together and wanted to determine when lesions would appear, if we could use PhotosynQ and the MultispeQ to detect it. So it was a two-part to find the non-visible impact of the plant health from the lesion, and then to see if there's that non-visible stress signal before we can actually visually detect it. And so this invisible lesion project occurred in growth chambers. We had inoculated plants, as well as some mock inoculated controls. Because it was an intense grid pattern, the amount of leaves is very small, so it's about 6 leaves. And after 13 days of the experiment, you end up having 45 lesioned areas that you can look at, and about 1,200 healthy areas of the leaf. So that kind of tells you the time that it takes to kind of get this kind of detection. And this is just a visual of a corn leaf. So this is how it occurs. This is the inoculation line. You just go through with a MultispeQ at each point up and down the leaf to try and get the location and that before the lesion occurs. So after getting all of this data back, I kind of looked at regression models to see which parameters were impacting the lesion, and so you can see that PhotosynQ is far and above kind of the best indicator for a lesion. User, unfortunately, was pretty high on the significance as well, which is good information if you're thinking of doing a in-field determination of an expensive fungicide cost. And then kind of typical SPAD, because you can visually see these, was important, as well as PAR and some temperature. Then I was wondering, okay, these parameters are important. How well could an algorithm classify if there's a lesion or not to build this kind of model? And it turns out it can't. It does a really bad job, which is odd because you would assume that visually you can see it. There's these very high measurements. However, it misclassifies about 40% of the time, which isn't great. And kind of led to another question of, okay, this isn't great, but if you could do this a couple days in advance, maybe, a 40% uncertainty is better than just plain guessing. So I'd like to see how soon you see these kind of lesion impacts from PhotosynQ. And unfortunately, it is both location specific and only occurs the day before. So if you make heat maps of these leaves kind of leading up to lesions, everything looks orangey and normal, following par, kind of as you would expect, until the day before, where you get these bright warning signals. So you can predict, it's just very specific and very quick to happen. And then if you go back and say, okay, in this location, will a lesion appear tomorrow? That classification is even worse. So just as you can assume from these project findings, they weren't great for this kind of idea of early detection of northern corn leaf blight. The areas are very location specific. The formation you can really only see a day before it happens with those parameters. And there's difficulty in models telling the difference between lesioned and healthy areas. So this foliar disease is not well suited. Kind of the cost benefit of going out and measuring doesn't add up to the next day you can see it. But it's good information. They weren't sure about the effective lesions, and they were really thinking that this could happen. So kind of knowing that it is a very small area is valid, is good information for their basic science questions. Yeah, and so I won't leave off on a totally devastating note. One of the nice things about PhotosynQ is that everybody can look at this data and they have their own questions. So a group that's trying to do aerial detection of northern leaf blight and is training cameras on that asked about the differences between lesion and non-lesion area, you know, what kind of stats could I give to help them out. And so I went back and looked at the differences and broke down by resistant and susceptible, which are up here. And kind of surprising, I found that the resistant line's actual lesion and damaged area is more photosynthetically active, significantly so. So when you kind of look back through this on a breeder's lens, it gets some very important ideas across that you can have, let's say, both of these leaves on the right look similar. If you had two lines that looked like that, you would both either be great or discard them, whereas their lesion and the actual photosynthesis and the grain filling that they can do could be very different. So just to throw some numbers and bars up there, we've got susceptible and resistant for phi and PQ. So high generally follows more damage we've seen in our models. And this lesion area, you can see it's about half. It's doing much better in that resistant line, actual damage area. And same with phi 2. So there's a big difference, and it kind of plays to when you kind of have this data out in the open and you can have different areas look at it, you get a lot of different results and interpretations. Thanks to Kramer Lab, just a couple people, thanks to all of the developers, and the Nelson Lab, along with Ag Bio Research, for hosting this today. And questions? Yes.
So maybe I missed it at the beginning of the presentation because I came in a little bit late, but so with all that classification stuff, are you using just one variable to classify?
So I used all of them,
Okay.
and then I narrowed it down, and some of the narrowing down was based on these right here. So these are the full list, and this is kind of an importance measure, so you can see phi and PQ is up there, genotype's quite low, so you can do that to narrow things down.
Okay.
So you said it wasn't necessarily that great at predicting lesions the next day or whatever, but do you think that the the MultispeQ device was still detecting real differences, or that might have been, or that means that something else was affecting the plant, or was it just like, were these inoculated, strictly inoculated plots, or could there have been something else going on with the plant that PhotosynQ was picking up that we just can't explain?
So kind of the nice part about this was it was a growth chamber, so it was a very sterile environment, specifically inoculated with that. And they were very careful because there were other experiments going on at the same time with the northern leaf blight. So they want to make sure for histopath and other reasons that it was as perfect as it could be.
Gotcha.
Yeah.
Just looks like it's really good at predicting when there wasn't going to be a lesion.
Yeah, it's great to tell them when it's healthy, so that's nice.
That aerate's very small, which is interesting. I'm trying to think.
Actually, I have, on one of your last slides, you showed that they're, the ones that are susceptible have higher PhotosynQ, if I remember correctly. Right, the ones that are susceptible they have high PhotosynQ, whereas the resistant ones have a lower.
Mm-hmm.
So and you said that they have even increased photosynthetic activity.
Yeah,
So what I was wondering is if by reducing the amount of quenching, you produce more reactive oxygen species that could help to form those lesions and to kind of like fight the fungi. So I wonder if has anyone looked into that, like kind of like what the defense response is? And so I mean that would explain why when you have less NPQ, and then this would help the plant to overcome the pathogen.
Yeah, I don't know the exact kind of defense mechanism for the pathway, so that's a possibility that those reactive oxygen species are kind of fighting it. I just don't know.
Even if you use it to induce apoptosis or something.
I'm not sure for leaf blight, but I know that programmed cell death caused by like just a massive relief of oxide burst within the cell is what kills it, and that is a part of fighting some diseases. I'm not sure if it's for blight.
Yeah, I think it'd be part of the response. It'd be a temporal spatial thing too on the leaf though, right? So I guess if you're talking about the part that's going to develop a lesion, then yeah, you'd probably be seeing those reactive oxygen species. And where that lesion's developing.
I just wonder if the resistant ones, what you observe there is kind of a build up before you're actually see
in
apoptosis.
In the resistant lines?
Yeah, when you're, at least from the naive view, I mean, you have the NPQ, the quenching functions in place to avoid those reactive oxygen species, right?
Mm-hmm.
So now you have a case where you actually want them.
Right.
So I'm just wondering if that's something you observe there. But I mean, maybe it's a little far-fetched. I don't know.
Well, it's probably just happening at very fine scales though, right?
Yeah.
Like the resistance is shutting down that fungus before it can sort of ramify through the leaf and cause a lot of lesions. So it'd be very localized, I would think. So whether you can measure that or not, I don't know. Yeah, but maybe that's part of what we're saying.
Okay.
So how much error do you think is associated with the fact that you're only measuring a small spot? Because can you see the lesion before you can't see it?
No, and if I could see the lesion, then there was a parameter for visual lesion.
So do you know if the effect of the infection would have a heterogeneous effect on the photosynthesis? Like you're only going to get high NPQ in the areas where you have an infection?
So that's what we were kind of trying to see, is if you do see kind of like from the inoculation site, that kind of gradual outward of like healthy appearance. We're kind of looking at the data, and like I said, looking at just the heat maps visually, you don't see that kind of trend like this is the infestation or this is the inoculation happening. It does seem pretty quiet until that snap.
So you're not measuring at the inoculation site, you're measuring at some distance from it?
I measured 6 centimeters below the inoculation site and some amount above.
How did you inoculate? Like with a droplet or an agar plug?
Okay.
Okay.
I know it is a specific line because they're able to make a mark at the inoculation site. Yeah. To my knowledge, spray had something to do with it.
Yeah, I probably spray Canidia on or something. So you can set up exactly where you want that lesion to form, right? If it's a Canidia spray, you can spray the whole leaf for that matter. You should get a lesion across the whole thing, I guess. If you've got conditions for infection.
Yeah, just contaminate your photosynthesis.
Yeah, that's right.
I had to make sure to like let it rest for a while, yeah.
So that's actually a good point. You did say user had like a significant
Yeah,
effect. Were the users using different devices?
So I used 2 devices the whole time and switched them off. However, when I brought somebody else in, that's when that user showed up. So it was someone else who was new to using the MultispeQ also having a difficulty.
So it seems to be user, not necessarily device.
Right.
Okay.
Yeah. Because the devices, I used 2 and they were used pretty consistently throughout the whole experiment across the leaves.
Cool.
Okay.
Thank you.
I have one last question
Yep.
about normal, instead of inoculated plants, normal infection in the field.
Yeah.
How many spots do you think you need to analyze to be able to get an idea if this thing really is resistant or not? Because inoculated is kind of cheating.
That's a good question. Yeah. That's a good question. So we do have a partner with an agronomy consulting firm in the summer, and they have some Northern Leaf Blight and some different lines. So I was thinking during the summer to kind of double check that to see if it plays out in the field and across a wider range of plants. Yeah.
Because when you're doing 10,000 plots, contaminating every leaf is not.
Right. It's not the best. Yeah.
Okay, I guess.
And I will briefly introduce Mitch.
You better. Great.
Yes.
Thank you.
So Mitch is a second-year PhD student in Marty Chilver's lab. He is interested in the genetics of plant pathogen interactions and the molecular basis of disease resistance. So the Chilver's lab uses a diverse set of methods to improve understanding of biology and genetics of host pathogen interactions. The lab's interested in using photosync to improve phenotyping and diagnostic activities. They work with fungal and oomocyte. Oomyces. Yes.
It's a tricky one to get the hang of. Yeah. Thank you for the introduction. My particular project is looking at soybeans and the disease called sudden death syndrome caused by a fungal pathogen, Fusarium virguliforme. So one of the things we were really interested in seeing if photosync could help us do is facilitate early detection of soybean SDS, and also whether or not photosync could give us a measure of root health without having to do destructive sampling. So I'll get into the biology a little bit of, oops, I gotta do it. Yeah. Okay, so Fusarium virguliforme is a fungal pathogen. It forms in culture, it'll form these water droplet looking things called sporodokia. And these are filled with asexual spores called conidia. I like to think of them as little smiley faces. They're smiling at me. And so these things will then germinate and find their roots and cause the first symptom of soybean SDS, which is the root rot symptom. So here we had an inoculated root. It's black and rotten, where compared to our control, it's nice and off-white color, nice and healthy. And once it establishes a really severe infection in the roots, it's thought to colonize deeper and deeper into the tissue and actually get to the vascular tissue, where it can then secrete phytotoxins that move up the xylem and into the leaves, causing this actually really pretty symptom of foliar interveinal chlorosis, the yellowing, and then necrosis. But the pathogen itself, Fusarium virguliforme, cannot be isolated from the foliar tissues. So it stays in the roots below the soil level. So then it can continue to grow there and form tough chlamydospores for overwintering, and inoculum can build up over time until you get fields that look like this, and these symptoms can really come on in a matter of 2 or 3 days. It's really why it's sudden death syndrome. All of a sudden, your plants, they seem healthy, and all of a sudden they look like that. And it is capable of causing up to 100% yield loss. So in Michigan, we have identified it in many counties since 2009. And for our PhotosynQ, we had set up a field project that we then incorporated PhotosynQ into down in Van Buren County in Decatur, Michigan. So the green star there is where the field plot was. And so the plot design looks a little bit like this. So current diagnostics require destructive sampling. I alluded to that before, where the first symptom, root rot, can really only be detected if you pull the plant out of the ground and look at the roots, and then either do an isolation or a qPCR assay to prove that the pathogen is there. So we did that. We did some of this destructive sampling, and at the same time, the PhotosynQ crew came out and measured photosynthesis levels at the same locations that I was taking destructive samplings at. And we wanted to see whether or not they correlated. So back to our main question, can PhotosynQ help facilitate early detection without destructive sampling? And also, extending on that, can it help predict the development of the foliar symptoms? So we did a grid sampling design where we had each box here is a 4-row plot, and I took destructive samplings from the outside rows. So I lettered those A and B are the outside rows of this first plot, and then we took 16 of those. So a grid sample design, and the PhotosynQ crew came behind us and did a very similar technique using up to 5 devices at growth stage V3. So I guess a quick reference, soybeans grow, these were the main stages we were interested in, V3, so the third trifoliate leaves were full and developed. At R1, the PhotosynQ crew came in again, where the flowers were just starting to be produced at the first nodes, and then at R5, where the seeds are almost mature enough to be touching each other, but not quite. That's when we took our second destructive sampling measures. So since it is a new technology, there was a little bit of technical difficulties trying to figure out how exactly the PhotosynQ crew should take the measurements and how many per plot. And we had done this experiment without PhotosynQ the year before, so our methods were pretty set in stone, but they had some flexibility. So at V3, what they did is they took measurements only in the morning, and they used 1 device per row. So in row A, where I took samples, they had 1 person go. In row B, and they were measuring 5 plants within that row, and only the uppermost middle trifoliate to try and get some consistency. But then at R1, the plants are a little bit bigger, and they did sampling in the morning and in the afternoon. They measured 1 plant from each, but they measured 3 different middle trifoliates from the low, middle, and top of the canopy. And so it's a new technology, and out in Decatur, the internet connection is not very good, so they were having some difficulties getting PhotosynQ to talk to their Android devices. But for the most part, we actually got a lot of data. So we had some tough technical difficulties the first time, but fewer the second time. But most importantly, the data seems to be telling us something. And now it's up to us to try and interpret that. So what I've got here is kind of intimidating. It's just a correlation matrix. So what I tried to do is group the PhotosynQ variables, phi NPQ, phi NO, NPQT, QL, LEF, and SPAD, all at growth stage V3, so their first sampling time point, and correlate them to my disease measurements, both at V3 and R5. So some of the takeaway things that I noticed is just that it seems like our PhotosynQ variables are correlating with themselves, and I don't know if that's expected or not. I don't know if it's a bad thing or not, but from my understanding of photosynthesis, you do expect some of that, especially the phi variables, phi 2, phi NPQ, and phi NO. They're kind of 3 different components of the same process. So you might see some inverse relationships there, which is fine. And then our variables are also correlating with themselves, but these are a little bit easier for me to understand biologically, where if we have disease index ratings at 3 different growth stages, right, they correlate because they're the same measurement just at different point in time. But the most interesting part is here, where those things interact. So at growth stage V3, the biggest thing that I noticed was this right here, that SPAD, LEF, and QL had a significant negative correlation. I know it's significant because the box is colored in. If it's not colored in, it's not significant at alpha of 0.05. So these are significant negative correlations to my V3 root rot ratings, which are on a scale from 0 to 100, just percent of the root that is rotten. So that's interesting. That might show that taking photosynthesis measures is reflective of root health with respect to root rot. So that's promising, that's interesting, and I hope that trend continues. Another trend we're starting to see is this significant negative relationship between V3 phi-NO and V3 phi-NPQ has a positive correlation with our R5 disease ratings. But that becomes even more prevalent at the R1 stage. We can see these 3 phi variables have this unique trend where phi-2 seems to be negatively correlated with disease development. Phi-NPQ seems to be positively correlated to disease development. And phi-NO, similar to phi-2, has a negative relationship with disease development. And the cool part about this, right, the previous slide we were comparing V3 measurements to V3 measurements, so it was kind of indicative of real-time root health. Whereas these were taken at R1 and we're comparing them to R5. So this actually indicates that it could be a useful predictive tool. If we take measurements at R1, we might be able to predict how much foliar SDS we will get a few growth stages later. So these correlation matrices are cool. They show the statistics, but another eye-catching way to show it is just show the actual representation of the data. So, like, we walk up and down this row to collect the data, up and down this row, and then we can use spatial interpolation, a spatial interpolation technique called kriging, to then create a visual heat map of the rest of the field. And I like these 3 variables in particular, root dry weight, R5, SDS, disease index, and phi-NPQ measurements at R1, because we can see here, right, the higher the root dry weight, the lower the disease was. So it shows that the disease is causing the roots to be rotten and go away. It reduces biomass. But then phi-NPQ, this map looks a lot like disease index. So it seems like anywhere where we might have a high phi-NPQ value, that's where foliar SDS is going to develop. I mean, these look very similar in my book. So that's promising to me. So take-home messages is that photosync to us seems like it can be representative of real-time root health based on the V3 data, and that it may have some predictive capacity as well, because the R1 values seem to correlate significantly to the R5 disease development ratings. So with that, I'd like to thank you guys for attending. Thank you, Marty, for the support, and Zach and others in my lab for some of the statistical expertise and help, and the PhotosynQ crew for taking the measurements. We had over 500 data points per growth stage, so it did a lot of work for us. Thanks. So, any questions then? Yeah.
I just have a general question.
Okay.
Because I'm seeing a lot of trends, and I'll even believe the trends. But my breeders will never believe those trends. Do you think anything could be done, or what should be done in order to bring those up into the 0.8, 0.9 sort of range?
You mean the correlation coefficients and whatnot? Yeah. That I'm not 100% sure how we can do that. So I do know, some of the people in the PhotosynQ lab, I think Greg has looked into trying to normalize for some of the effects that we can get rid of, like user effects that you were seeing. He had done some data, some correlations just with user to try and eliminate that as having a confounding effect. And some of the other things too, like time of day can have an effect, right? Or PAR is another one that can have an effect that isn't necessarily representing the real biology out there. So I think with some more of the optimization of those types of data manipulations can really boost those numbers. But we're still not really sure how many of those are appropriate to normalize for. So we're taking steps in that direction, but we're trying to be careful about it. Yeah.
I thought it was interesting to see that even with a slight correlation, the traits that you would hypothesize would be associated with disease, NPQ specifically, it's nice to see that they both sort of rise together to get more disease. You get more NPQ, which is the plant like, oh, I can't deal with all this light that I have now because there's something wrong with me. I need to dissipate it. And it makes sense that it'd be caused by disease.
Yeah. So, and the foliar symptoms are rather, impressive, and we can see those, but those typically happen later in the season, and so it could be just a sense that that photosynthesis is picking them up earlier than we can. But it could also represent the plant's physiology too, right? I was trying to understand this earlier this week. So PhotosynQ, correct me if I'm wrong, is kind of a measure of photosynthesis downregulation.
So it's the fraction of light that's actively being quenched.
So
you're
that's not going towards carbon fixation.
Yeah, it's actively being quenched, so the plant is actively shedding.
Right. So if you have a lot of that quenching, a lot of that's not going towards photosynthesis, who knows where it's going, but it's not going towards photosynthesis, which is important for plant health. That seems to be where the disease is letting up. So I think the biology does make sense. It's a little hard to wrap your head around for me because I'm not quite as familiar with the photosynthesis.
Well, let's say that in this case your root tissue is like winter major sinks, right? You have nowhere now to put your fixed carbon. So what happens is you back up photosynthesis. And so if you do that, then you can't use all the light energy that you're absorbing. And since it can destroy your leaf, the thing you want to do is get rid of it. So you engage the active conjugate, for example. So that's one way I guess you could imagine these things happen.
Yeah. So there's a theory out there from the side of the pathogen that different isolates are going to have different virulence levels, and since it's kind of a two-step disease symptom, root rot and foliar symptoms, there's this thought out there that some isolates are really good root rotters but not good toxin producers, and the opposite may be true. That might be a cool thing to do, is try and identify isolates that are good root rotters but not good toxin producers, and vice versa, to see if the root being a sink for sugars still holds true. Or still makes sense then in this case. Right? Like it's still downregulating because it doesn't have a place to store the sugars or use the sugars.
I'm just going to ask a general question about the pathogen. Is there an advantage to the pathogen for its plant host to die? Like for it to essentially deliberately, but like to send that toxin into the plant to kill the photochronic tissue?
Yeah, so it's a hemibiotrope. I think it'd be advantageous because it can get more nutrients that way, and it's also pretty good at overwintering. And there's also a study out there that shows it's got a pretty high host range, a pretty wide host range. So maybe it doesn't really care that the plant is dying because it can overwinter. It forms the tough chlamydospores, and the next year, even if you don't plant soybeans, it'll make do with whatever host you put there. But what's interesting is that these foliar symptoms seem to only show up in soybeans and in dry beans in some of our greenhouse experiments.
So it's a generalist sort of pathogen.
Yeah. Although we're not quite sure the impact it's having on the other crops just yet.
So would there be any practical advantage to being able to protect them, or when is there any sort of treatment you can do for?
Good question. Unfortunately, there's not a whole lot you can do right now. There's no way to treat an active infection. There are a few chemistries out there that are being tested for their seed treatment ability to prevent infection. Bayer has a product that we've tested in our lab. Syngenta markets a product that some people have had some success with. I don't know if we've used it a whole lot in our lab.
We just screened it for the first time last year.
Oh, okay, great. So we're still testing some of those chemistries, but you could also argue that early detection would be, this would have a practical advantage for early detection, because if it's early enough, you may be able to replant with something else. If you think SDS is going to be that big of a problem and that yield limiting, maybe you can grow something else. Dry beans require a shorter growing season than soybeans do.
So how many weeks post-planting is that? Because I mean, that's always the kind of the holy grail, is to be able to make a replant decision within a week or two.
So can you rephrase your question for me?
Yeah, how late post-planting?
Can we detect the symptoms?
Yeah.
I don't think we can claim we can detect it.
Well, in this experiment, how early is V3?
Well, so V3, it was about a month after planting, so it's about 4 weeks. But the part of the study that I didn't go into was the original idea behind the whole project was whether or not we could make disease predictions just from the soil before the seeds are in the ground. And that seems to be promising as well. So maybe in collaboration with the PhotosynQ stuff, we can be able to predict it even better.
So that was kind of my question, like, how long does it take for the SDS to form the overwintering spores? Because it forms it right away, or if you can, kill the plants early enough to where it doesn't quite form those spores yet, could you reduce the infection in your field if you plant something else?
No, I don't think so. And most of the spores are probably produced later, right, when the roots are rotting and everything. We see the Sporodokia, but that's not a viable alternative. Like if you go out and spray it with a herbicide or rip the field up, the tillage, it's just going to feast on all that dead organic matter anyway. So it's a really difficult pathogen to manage in that sense. I'm really just excited about the potential to predict root rot, right, without destructively pulling up plants and whatnot. So the whole pulling up plants is a lot of work. So if you had this PhotosynQ is a lot less work than putting a shovel on the ground. So that's where I see the real advantage. Screening germplasm too, right? The breeders can screen potentially, if it works that way, you could screen for root rot, in a breeding sort of sense too, and capture that.
Even if it wasn't early detection, it would still prevent you from having to put a shovel in the ground.
Right, which is huge, right? It's a lot of labour saving.
Even for chemistry development.
Yeah, exactly.
That's why there's nothing out there.
Right, absolutely.
And honestly, shoveling the ground's only half the story too. And our field plot is so far away that we have to then take them back to campus and wash the roots, rate each root, and then dry it for 2 days to take dry root measurements, and then grind the tissue, do qPCR to do like a real true diagnostic test. So this could speed that process up really by a lot. Any other questions? Were we slotted to the floor?
Yeah, you guys did great.
I have a final. So you see this more as for breeders, less as a management strategy? Is that what this?
Because there are no really effective management strategies. I think it seems to be more promising for the breeders at this point in time, but we certainly can't rule out the usefulness as a management tool.
Yeah.
So how much sampling do you think you're going to need to do to convince a farmer that you're coming up and saying his soil is full of Fusarium and not to plant beans?
Yeah, so that's a good question. The sampling grid design we've got is, we've done 96 samples in what's like a 100 by 30 foot total plot. So that's pretty intense sampling for a fairly small.
I don't see it happening on 1,000 acres.
Yeah, So realistically, maybe it's not.
It's the
as
same
useful right
problem. It's analogous to soybean cyst nematode.
Yeah, I was gonna say, it's the same things they have with cysts.
So, yeah, I mean, they're never going to be 100% perfect, but if you can do some prediction, then that's better than nothing.
But at least they can get low though, because they'll send them out for counts.
Right, but we can use qPCR. So he's talking about another project where you can use real-time PCR to quantify the fungus in the soil, which he didn't have slides on, but yeah, that's what he's working on in the field as well. And that seems to predict at least 1 year's data, right? Looks pretty good within a field. We're just doing within a field at the moment too, just to validate the procedure before we take it to multiple fields.
Yeah, so we have 2 years worth of data that he's talking about where we have soil samples and quantifying the fungus, not quite counting like nematodes, but qPCR is a quantitative method. From the first year we had 24 points in our grid sampling design. Here we have 96, and more sampling improves our predictive power, but even at a low resolution, low scale sampling, we do have some significant correlations. So if a farmer knows his fields, he might be able to do some targeted sampling and still get predictive power and not have to sample the whole field intensively.
Could you use it as like an early step? So, I mean, now you go through your field and you take samples for each plot, right? So when you see that some of the parameters are off, maybe they're not like powerful enough or good enough to like convince you to just employ your field again. But then you could go and start sampling, maybe revive some plants and look at the roots. Would that be already beneficial? I mean,
probably. Yeah.
Absolutely.
Yeah.
Yeah, if you know.
it's not a perfect predictor. It might never be.
Right.
But if it could like tell you that something is unusual,
right.
And I mean there are other pests out there, right? They might have a similar effect that we see in terms of the photosynthetic parameters.
Yeah. So
but they might allow us to then say, okay, now it's something wrong, and we should take a closer look.
Absolutely. So that would sort of couple in with some of the other stuff we've been talking to you guys about, Dave. And Amy and Brad are talking in the other session, right? So using the PhotosynQ platform to do some sort of colorimetric diagnostic assay in the field. So Amy's been playing with that with Vanjie and looks promising. But yeah, absolutely. If you could take a photosynthetic measurement and know that, yeah, we've got root rot, okay, let's pull up a plant, let's do a quick rough grind in the field of the DNA of the root, get some DNA out, run the colorimetric test and put it through the PhotosynQ device too, right? Yeah. Could totally go together.
If you were in this part of the field, PhotosynQ was telling you, don't worry about it, it's fine. Everything's good here. We could have left that part of the plot alone. If you were down here, PhotosynQ is telling us, wow, something's wrong here. Right? We go and look at the roots, and the dry weight is significantly down. Now, sure enough, it's probably because of rot. It could be, nematode feeding has just devastated it. Yeah. Definitely, it could be a proxy of where to take your destructive samples.
How confounding do you think data would get if you're looking at a plot system that has germplasm of varying resistance? Get it?
Quite confounding, probably. So this plot design was using one seed type. Our variable of interest, if you will, was the soil. So we tried to normalize everything else. We used the same seed germplasm, the same spacing, everything. So yeah, we haven't extended this to other genetic backgrounds yet. Because that's one thing that breeders are trying to do is develop germplasm that has resistance to this stuff. But we haven't extended our PhotosynQ project to involve that yet.
Because that'll be a segregating population, and they'll be testing all kinds of different things in it.
Yep. And probably your baseline photosynthesis measurements will be wonky anyway. You probably need some photosync data on them growing in the greenhouse in a healthy environment just to know what's normal.
But I'll have the replicates to know what normal is for that particular variety is. It's not quite a variety yet. This is a line. That's the one thing that we will do is we go out in big numbers, so we get the statistics behind it. But I could see it, you nailed it, I could see the baselines changing at the same time you're trying to get that change on top of it.
So in one cultivar, yeah, we can see differences, so that's great, that's proof of concept then.
Yep.
Thank you.